Hello, dear colleagues. Thank you for a great honor to present my country, to present my institute with results of our work, vitrification of tissue-engineered constructs based on mesenchymal stromal cells. Tissue-engineered constructs based on multipotential mesenchymal stromal cells can be scaffold-free like spheroids or scaffold-based. There are a lot of approaches to get tissue-engineered constructs. Different types provide different environment for cells and support different realization of different properties like enhanced paracrine potential, enhanced stemness, immunoisolation, or ability to grow cell-to-cell and cell-to-matrix contracts and with in vivo-like environment. And practically, TECs are widely used as tissue models in biological researches, in drug development, as tissue substitutes, in regenerative medicine, in immunomodulation therapy. Any practical application of TEC require effective and simple preservation techniques. Storage under culture condition and hyperthermic storage can provide only short-term storage. Conventional slow cooling methods support long-term storage, but such methods are based on slow cooling with controlled ice crystal formation, but such ice crystals can destroy scaffold structure. Vitrification is based on glass-like solidification of whole system and exclude crystallization process, so such approach are considered as promising for long-term storage of tissue-engineered constructs. At the same time, on practice, TECs vitrification face a set of challenges, and first it linked with type of container. Up to now, most of vitrification protocols have been developed for open containers, for clinically applicable tissue-engineered products. Closed containers with relative large volume should be applied. At the same time, it decreases levels of rates of cooling and warming and affect success of vitrification process. Because vitrification system is set of interrelated variables which involve vitrification solution composition and properties, type of container, technological steps, and properties of biological sample. If one of parameters is changed, it may need to change other criteria. TEC are relatively complex object for vitrification, and set of parameters should be controlled like CPA toxicity, saturation process, cooling and warming rates. At the same time, thermomechanical stresses and cracking should be managed, and devitrification at warming stage should be prevented. Here we aimed to develop the composition of vitrification solution and steps of vitrification procedure in standard plastic cryovials to prevent the development of crystallization, vitreous phase cracking, and to preserve cell viability for two types of TECs with MSCs, alginate microspheres and macroporous scaffolds-based TECs. First part of our study involved choice of vitrification solution composition, development of vitrification procedure with model object MSC suspension, customization of vitrification procedure for MSCs encapsulated in IMS. To develop vitrification solution composition, we analyzed mixture of cryoprotectants, dimethyl sulfoxide, ethylene glycol, propanediol, and sucrose. That's why we called our mixtures DEPs. Visual analysis help us to choose compositions which were able to prevent visual signs of crystallization, and then chosen solutions were analyzed with help of DSCs, DSC, differentiational scanning colorimetry to analyze phase behavior. Two solutions were chosen with help of visual screening analysis. depths 10-20-20 with 10% of dimethyl sulfoxide and with 20% of ethylene glycol and propandiol and 0.5 mole of sucrose, and depths 10-15-15, 10% of DMCO, 15% of ethylene glycol and propandiol, and 1 mole of sucrose. Phase behavior of both solutions reflects that actually during cooling we get a process of vitrification. So such during warming we observed glass transition, crystallization and ice melting, and first solution revealed more beta glass forming tendencies. That's why was chosen for subsequent experiments. Then with depths 10-20-20 we developed vitrification procedure steps for cell suspension and then for MSCs in alginate microspheres. Vitrification procedure involved two-step exposure, then cooling by direct plunging into liquid nitrogen, sowing on water bath, and then we analyzed cell viability of cell exposure to solution and devitrified. After exposure and after vitrification, viability index is decreased, but stay sufficiently high. And what was even more important, after vitrification, cells were able to attach to tissue culture plastic, showed normal morphology, was able for growth to form monolayer. After culture in osteogenic and adipogenic medium, cells showed ability for induced differentiation. This protocol was applied for cells encapsulated in alginate microspheres, but results were poor, only 5% of viability. So we decided to modify protocol with increase of exposure step duration, and this helped us to get 75% of viability and 53% of metabolic activity, and moreover, devitrified cells were able for differentiation in osteogenic and adipogenic directions, both inside of microspheres or in 2D culture after dissolving of AMS and extraction of MSCs with help of dissolving in citrate solution. After that we performed second part of our study, choice of possibility to control crystallization in macroporous scaffolds, choice of possibility to control cracking, and then we applied modified protocols for macroporous scaffolds based tissue engineered constructs with MSCs and perform vitrification of such constructs. We used depths 10-20-20 and we use the same approach of prolongation of exposure steps, but unfortunately even prolongation of saturation of scaffold, cell-free scaffold with solution for 50 up to 15 minutes, this didn't help us to prevent crystallization inside of scaffold. That's why we decided to modify solution, and we have increased DMCO concentration and ethylene glycol. DMCO increase affected ability to prevent crystallization inside of scaffold more greatly than increase of ethylene glycol concentration. Then we applied another approach, addition of synthetic ice blocker, polyvinyl alcohol, and such combination increased dimethyl sulfoxide up to 15%, depths 15-20-20, with added 1% of polyvinyl alcohol helped us to prevent visual signs of development of visual signs of crystallization inside of scaffold. Modified solutions were tested on MSC suspension, and also in this experiment we modeled two types of saturation approaches: perfusion method and concentration gradient transfer, which is more popular, but our results showed that perfusion method is more mild for cells and helped to get high indexes of viability and metabolic activity compared to concentration gradient transfer. So modified solution was tolerated with cells and helped perform successful vitrification of MSC suspension. Next step of our study was choice of possibility to control vitreous phase cracking. Based on thermal characteristics of solution obtained with help of DSCs, we developed two-step cooling, rapid cooling at first step and slow cooling at second step, to stop development of thermomechanical stresses. At this picture we can compare one-step cooling and we see cracking, one-step warming exacerbate this cracking. At the same time, two-step cooling and subsequent two-step warming completely prevent cracking. Modified protocol were applied for vitrification of macroporous cryogel scaffold seeded with MSCs, seeded and cultured MSCs. We can see that this cell are fibroblast-like and make a lot of contacts. After exposure, almost all cells are still alive, but after vitrification, only few cells are alive. So such data indicates that cell death during vitrification is linked with development of visually undetectable intracellular crystallization, which is possibly supported by formation of cell-to-cell and cell-to-matrix contacts. So we need to search some another approaches to solve this challenge. To sum up our results, I should say that vitrification system included multi-component vitreous solution, closed type container, human adult MSCs, and two-step exposure was developed. This vitrification system provided successful cryopreservation of MSCs in suspension and MSCs encapsulated in alginate microspheres in relatively large volume, up to 1 milliliters. Set of conditions necessary for vitrification outside and inside of macroporous scaffolds without cracking is determined as a result of our work. Modified vitrification protocol provided high viability rates of MSCs in suspension, but should be further improved for vitrification of MSCs in macroporous scaffold. For success, additional approaches should be engaged. Possible approaches for future research involve ice-binding proteins, inhibitors of ice nucleation, non-toxic high-viscosity molecules addition, and so on. And I hope we will manage to solve this problem. I would like to acknowledge all staffs of my department of cryobiochemistry, and I hope to meet all of these people soon in Ukraine. Thank you for attention, and please stand with Ukraine and help us to stop Russian terror. Thank you for attention.